Hello my dear friends, in this video we are going to see clearly about what is sliding filament theory. Yes, so let's go into the topic. First we have to know about the microstructure of skeletal muscle, which is nothing but the overall muscle is being on a cross section. Inside that we have a muscle cells. So that muscle cells we also call it as muscle fibers because it looks like a fiber. Inside that muscle fiber we have myofibrils. So inside that myofibril we have sarcomere. So what does a sarcomere consist of? The sarcomere or inside the myofibril it has the actin and myosin. Actin is the thin filament and myosin is the thick filament. So, myosin filament is thick in nature, have long rod shaped tail with two globular heads. So, the heads will form the cross bridges. So, here we have the heads. So, this is the myosin molecule. Here we have the head and this is the tail. So, this head and tail. So, this is the 3D model. So, we have the head as well as the trail. Head and trail. This is myosin. Coming to the next, actin. Actin is thin filament. So we have tropomyosin. So three parts are there in the actin. We have the F actin, tropomyosin and troponin. Tropomyosin is rigid rod-shaped protein which lives in grooves on either side of the actin. Troponin is a complex of three globular proteins. So this is the picture of the actin. So we have the actin. On that actin, we have the tropomyosin, which is like a thread-like structure. On the tropomyosin, we have the troponin. So, this bigger circular structures are the F actin. So, actin, so which we also call it as functional actin. On that, so on the in the grooves we have the tropomyosin. On the tropomyosin, we have the troponin. Coming to the next, this is the clear picture about the actin and myosin. So once again, actin is thin filament, myosin is thick filament. So we have the actin filament. So this is F-actin, we also call it as functional actin, this blue color part. And this is tropomyosin, which lives in the grooves of actin. And on this tropomyosin, we have troponin. So this is the troponin. And this is the myosin filament. This myosin filaments will have the head and tail. So these are the heads. So this is the tail part. So myosin tail and myosin head. And this is whole thing we call it as myosin filament. And here we have the Z line. So what is a Z line, H line? In the next slide we are going to see. So next, let's see about the lines. So we have the Z line. So here to here. So this is one Z line and this is another Z line. The space, the part between the two Z lines, we call it a sarcomere. So this is the entire sarcomere, the part between the two Z lines. Okay. And next, the center line we call it as M line because only the myosin filaments are occupied in this line. Because of that, they call it as M line. And we have the thick filaments, which is nothing but myosin, and the thin filaments, which are nothing but actin. So actin and myosin. So here we have, and again we have one more thing called A band and I band. A band and I band. So A band means anisotropic band. I band means isotropic band. Okay. Through I band the light can pass. Through A band the light cannot pass. So the center band we call it as A band and this I band. So in the I band we have only the actin. But in the A band we have both actin as well as myosin, which is thick in nature. So A band and I band. And the center line we call it as M line and the zone we call it as H zone where there is an overlapping of actin and myosin will take place. So H zone or Henson's zone. So this whole thing we call it as H zone. Sorry, there is only myosin will be there in the H zone also. H zone or Henson's zone. So during contraction this H zone will not be there because so the overlapping of actin and myosin will decreases the H zone. So these are about the lines. Once again, we have the Z line and the sarcomere. Between this we call it a sarcomere. We have the M line and we have the H zone, which is we are nothing but Henson's zone, and we have the A band, and either side of the A band we have the I bands. Okay. Next, structures involved. We have the myofibril, nothing but a cylindrical organelle running the length of the muscle fiber containing actin and myosin. We call it as myofibril. Sarcomere. the functional unit of the myofibril, divided into I band, A band, and H zones. Actin, a thin contractile protein filament containing the active or binding sites. Myosin, a thick contractile protein filament which produces known as myosin heads. Next, tropomyosin, an actin binding protein which regulates the muscle contraction. Troponin, a complex of three proteins attached to the tropomyosin, we call it as troponin. So this is the structure of the skeletal muscle. Initially we have the muscle. Inside this is the whole muscle. Inside the muscle we have the muscle fiber. Inside this muscle fiber we have the myofibrils. So this whole thing is skeletal muscle fiber. So this is one cell of the skeletal muscle. So this is the cell of the skeletal muscle. Inside the cell we have many myofibrils. So this is the illustration of one myofibril. Coming to the sarcoplasmic reticulum on transverse tubules. So the endoplasmic reticulum present inside the muscle fiber, we call it as sarcoplasmic reticulum, which is being arranged in a form of longitudinal tubules and transverse tubules. Inside the transverse tubules, more amount of calcium is being deposited. Because of that, it is highly important. So inside the sarcoplasmic reticulum only we have the calcium. It is important for sliding filament theory. Okay, excitation, contraction, and coupling. So the depolarization of motor end plate, we call it as excitation, is coupled to muscle contraction. So overview. Action potential travels down the transverse tubule and causes the release of calcium from sarcoplasmic reticulum. Now the calcium will bind to troponin and causes the position change in the tropomyosin, now exposing the active site on the actin. Now the strong binding state formed between the actin and myosin. Now the contraction will occur. Coming to the muscle contraction, the process of muscle contraction can be divided into 5 sections. One is excitation, a nerve impulse arriving at the neuromuscular junction which causes a release of chemical called acetylcholine into the synaptic cleft. The presence of the acetylcholine causes the calcium to be released from sarcoplasmic reticulum. In the presence of high concentration of calcium, the calcium binds to troponin, changing its shape. This moves the tropomyosin away from the active site of the actin to expose the uncovered sites. Therefore, myosin filaments can now attach it to the actin, forming a cross bridge. So this is the tropomyosin. And we have the troponin, and this is actin. When the calcium from the sarcoplasmic reticulum, first it will come and attaches to this troponin. So this troponin will causes the tropomyosin. Along with the tropomyosin, it will move away from the active site. Now the active site of the actin will be free for the attachment of myosin head. So then the cross bridges is being formed. The myosin heads bind to the actin, tilts the forcing the actin to move towards the myosin that requires the ATP energy. When the ATP molecule binds to the myosin head, the myosin detaches from the actin and the cross bridge is broken. When the actin is then broken down, the myosin head can again attaches to actin binding site further along the actin filament and repeat the power stroke. So the power stroke is nothing but very simple, like oars of the boat. How the oars of the boat will run the boat, so the same will be there. So initially this myosin head will attaches. First, initially the action potential, when it reaches to the nerve terminal, the acetylcholine will attaches to motor end plate. So later the calcium, which causes the calcium release from the sarcoplasmic reticulum, that calcium will attaches on this troponin. Now this troponin will pull the tropomyosin away from the active site. Now the active site is exposed. So now the myosin head will attaches to the actin. So this will form the cross bridge. So after cross bridge formation, it has to move. So the actin has to be moved. So to that energy is needed. For that energy, ATP. So the ATP will come and attaches to the myosin head, and now it breaks the bridge. So after breaking the bridge, the ATP will divide into ADP plus PI in the myosin head, and now again reattaches to the actin. So this we call it as power stroke. So the energy releases produces a cross bridge movement, which we call it as power stroke. So the new ATP attaches to the myosin cross bridge to dissociate from the actin. When the ATP comes to the myosin head, then only it will detach, or else it will not detach. So in rigor mortis, after death, each and every muscle of the body will stiff because of this case, because the ATP will not come to attaches to the myosin head. So there is no any breakage of cross bridge. So the muscle will become very stiff. So this is the illustration of the thick filament and thin filament. So the ATP binds to the myosin head, which is releases from the actin filament. When the ATP attaches only, then it will get released. Hydrolysis of ATP cocks the myosin head. Means cocks means, so it will flip the myosin head for the power stroke. So the myosin head attaches to the actin binding site with the help of calcium. So the power stroke slides the actin thin filament. This process of muscle contraction can last for long as there is enough ATP and calcium stores. Once the impulse stops, the calcium is pumped back to the sarcoplasmic reticulum and actin returns to its resting position, causing the muscle to lengthen and relax. So during contraction, neither myosin nor actin filament changes its length. So its length never be changed. They slide past from one another. Only sliding will happen. They will not shorten and lengthen. A band remains the same, but the I band will decreases. This is the A band. Okay. So the A band will remains the same, but the I band will decreases. Here if you see means the I band decreased. And here there is completely loss of the I band. Only the A band is there. So the H zone also decreases on construction. Only effect is for the H zone and for A band. So not for the I band. So the A band remains the same. The I band and the H zone will decrease. So this is the state when the sarcomere is relaxed. So this is the picture. Everything is normal. And this is partially contracted place. And next, sarcomere when it is completely contracted. There is no any I band and H zone. So this is all about the sliding filament theory. So please do subscribe to my channel for more biology videos. Thank you, my dear.